ఆ, ఆ. ఓకే, ఒక్కసారి ఇది షేర్ చేద్దాం, ఒక్క నిమిషం. ఆడియో అది ఉండాలి.
Good morning, sir.
గుడ్ మార్నింగ్.
Okay, sir.
ఒక్క నిమిషం.
Okay, okay, sir. You are audible, sir. Sir, screen share చేస్తే sir.
1 minute.
Okay.
చూడమ్మా, స్క్రీన్ షేర్ అవుతుందిగా.
Uh,
ఓకే అమ్మా.
yes, sir. Yes, sir. Screen share అవుతుంది, sir.
ఓకే, వాయిస్ కూడా వినబడుతుంది కదా క్లియర్
Yes,
గా.
sir. Yes, sir. Audible, sir.
ఓకే ఓకే.
Yes,
నేను
sir.
క్లోజ్ చేసి మళ్ళీ వచ్చాను.
Okay, sir. Okay, sir. Okay.
ఓకే.
Okay. Okay. Okay, sir. Okay.
ఓకే. హలో. గుడ్ గుడ్ గుడ్.
I request all the participants to mute themselves.
नाही 4 तासच पकडायचे कारण त्याला ह्याला लावायचं नाही जे मी दिलं ना तेच लावा त्याबरोबर
Hello. Good morning, sir. Can I start the session, sir? Principal, sir.
Good morning. You can start the session.
Okay, sir.
Sai Kishore Gadev.
Good morning, sir.
Good morning, Sai Kishore Gadev. How are you?
Fine, sir. Good morning, sir.
Good morning, and thank you. Rashid Gadev, you can start.
Okay,
Sir
sir.
is there.
Good morning, everyone. I, Sheikh, I, Sheikh Rashid, Assistant Professor and Convener of this program, thrilled to welcome you all to second day of our 1 week online refresher program, Navigating the Future of Pharmaceutics, Addressing the Challenges and Seizing the Opportunities. Organized by VV Institute of Pharmaceutical Sciences, Gudlavalleru Post, Krishna District, in association with APTI AP State Branch. As we embark on this journey together, we will explore the dynamic landscape of pharmaceutics. We will delve into the latest advancements, discuss emerging challenges, and identify the innovative solutions to shape the future of our field. Let's make this most of this session as we collectively strive to address and understand the complex issues facing by our industry and unlock the new opportunities for growth and innovation. Now I request patron, Professor and Principal Dr. A. Lakshman Rao Garu, VV Institute of Pharmaceutical Sciences, Gudlavalleru Post, Krishna District, to speak a few words about today's session.
Okay, thank you, Rashid Gadev. Eminent speaker of the today's session and senior academician, Dr. V. Sai Kishore Garu, Professor and Head Department of Pharmaceutics, Bapatla College of Pharmacy, Bapatla, Andhra Pradesh. Teacher participants from various pharmacy colleges, a very pleasant morning to one and all for the second day of 1 week online refresher program on navigating the future of pharmaceutics, addressing challenges and seizing opportunities, organized by VV Institute of Pharmaceutical Sciences, Gudlavalleru, in association with APTI AP State Branch. In day 2, Dr. V. Sai Kishore Garu will deliver a lecture on regulatory considerations for new drug development process. Regulatory considerations in the new drug development process are critical mainly for ensuring the safety and efficacy of the drug molecules. And developers must navigate a complex landscape of regulations set by authorities like FDA, by the by United States of America, and also European Medical Agency in Europe. And this includes conducting the preclinical trials and followed by the well-designed clinical trials that comply with good clinical practice, that is GCP standards. Submissions for investigational new drug applications, that is IND, and new drug applications like NDA must include comprehensive data on the drug's pharmacokinetics, toxicology, and clinical outcomes. Additionally, ongoing post-marketing surveillance is also very much essential component to monitor the long-term effects of the drugs and also to ensure the continued compliance with regulatory standards, making a thorough understanding of these requirements that are very much vital for successful drug development. With these few remarks for the today's talk, I welcome Dr. V. Sai Kishore Garu, and once again, on behalf of management, principal, and staff and students, we are very much thankful to Dr. V. Sai Kishore Garu. He is a very good friend to me, and he is a very senior academician. He has wrote several books also. He is having very sound knowledge on the drug regulatory affairs. So he is one of the pioneers in the drug regulatory affairs in Andhra Pradesh. Okay, we are very much fortunate to have that type of the senior academician for our refresher program. Thank you, Sai Kishore Garu. Now I hand over to Mr. S.K. Rashid, convener of refresher program, to introduce the today's speaker to the participants. Over to Rashid Garu.
Thank you, Lakshman sir.
Thank you, sir. We are grateful for your motivating and encouraging words. Now let me introduce you speaker of day 2 of 1 week online refresher program on navigating the future of pharmaceutics and addressing the challenges and seizing opportunities. Dr. V. Sai Kishore sir is a seasoned academician and a visionary researcher with a PhD in pharmaceutics. Currently serves as an head of departments of pharmaceutics at Bapatla College of Pharmacy, Bapatla, Andhra Pradesh. With a rich teaching experience spanning over 19 years, he has made significant contributions to the field of pharmaceutics. He has prolific publication record with 131 research publications published in esteemed national and international journals. He has guided 61 MPharm students and currently mentoring 5 doctoral candidates. A dedicated member of APTI and PCI, he has actively participated in various national and international conferences. Sharing his research insights, his doctoral research focused on design and development of innovative transdermal films for antihypertensive drugs. He has also contributed to improving the quality of the technical education in Andhra Pradesh by participating in development of e-lessons for the Department of Technical Education. Dr. Sai Kishore Garu's academics excellence is further evidenced by his authorship of 3 books: Novel Drug Delivery System and Regulatory Affairs, Drug Regulatory Affairs, and Pharma in Spectrum. His achievements have been recognized with prestigious awards, including the Association Fellow Award from Andhra Pradesh Academy of Sciences in 2017 and the Best Young Researcher Award from the International Innovative Research Foundation in 2020. He also secured second prize in cash award of 10,000 rupees at 18th APTI National Conference, a testament to his outstanding research contributions. With this brief introduction, I would like to hand over the session to our eminent speaker, Dr. V. Sai Kishore Garu.
Thank you, Rashid sir. My name is Dr. V. Sai Kishore. I am working as a professor and HOD in Department of Pharmaceutics, Bapatla College of Pharmacy. Today, I wish to give seminar on regulatory considerations in new drug development process. Sir, screen is visible, sir?
Yes, sir, the screen is visible, sir.
Now it is in title slide, Sai Kishore Garu.
What, sir?
Now it is in first slide.
Okay. One minute. First,
Okay.
good morning to one and one and all. Now, coming to the regulatory consideration. Regulatory considerations were developed from the desire of government to protect the public health by controlling the quality, safety, and efficacy of the products in the areas including pharmaceuticals, veterinary products, and cosmetics. Coming to the regulatory guidelines, long back onwards, there is a demand for the clinical investigation to assess whether a drug is of value in the treatment of disease or not. Coming to the regulatory considerations, from long back onwards, there is a demand, but there is no written guidelines till mid of 19th century. In 14th century, Paracelsus, he told that all things are poisonous and there is nothing that is harmless. The dose alone decides that something is no poison. In 17th century, William Withering also given one quote that poison in small doses are the best medicines and useful medicines in too large doses are poisonous. In 18th century, the Napoleon Bonaparte, he also told that I don't want two diseases, one nature made and one doctor made. Even though there are several representation, but there is no written laws for the governing the profession of pharmacy in India throughout abroad. But in mid of 19th century, due to the thalidomide tragedy, in between 1950 to 1960, thousands of children are born with congenital malformation in Europe, Canada, and Latin America. The drug thalidomide was prescribed to the women in early pregnancy to overcome the unpleasantness of morning sickness. Pre-marketing test in 300 individuals did not alert the investigators about its toxic effects and side effects. This tragedy prompted the US authorities to the concept that drugs need to be proven safety as well as treating the disease. In mid of 19th century, in the decade 1950, several children born with congenital, given there is a need for certain rules and regulations for governing the profession of pharmacy. This for this tragedy, first respondent is US FDA, United States Foods and Drugs Administration. They framed certain guidelines. Those guidelines are the drug need to be proven safety as well as treating the disease. Here, the regulatory considerations main intention is the drugs to be proven to be safety as well as treating the disease. Here, treating the disease means efficacy. Efficacy of the drug is secondary. The safety of the drug is the first priority. If you observe the drug development process, there are 6 steps: basic research, disease discovery, drug discovery, preclinical development, clinical trials. Next, it's development, manufacturing, and quality control laboratories testing. But in those, till the basic research to drug discovery, there is no need of any regulations. But once, if any drug identified as a promising candidate, it should be proven to be safe one by using the GLP, by following the GLP guidelines. GLP means good laboratory practices guidelines for preclinical trials, for conducting the clinical trials, GCP, and for the development of manufacturing, we will follow the GMP, good manufacturing practices guidelines. Here, the drug can be obtained from any sources. Here, there will be no need of any restrictions or any conditions, any rules and regulations. We can obtain the drug from any sources, whether it may be plant or animal or mineral or marine or semi-synthetic or synthetic sources or microbiological sources, or by using the recombinant DNA technology. From any source, we can obtain the drug. But once the drug is identified, in the discovery stage involves assessment the chemical properties of the new drug as well as to determine the steps for synthesis and purification. We will look into its isolation, synthesis and purification and isolation of the API, active pharmaceutical ingredient. Once, if you identify the API, then we will go for the systematic review, screening of the new molecule by following the preclinical trials and clinical trials. Here, the first step, after a compound has been identified as a promising, it enters a rigorous testing and evaluation stage is known as preclinical testing. This preclinical testing is also called as the non-clinical trials. It is a stage of research. It can begin and during which the important feasibility, interactive testing and safety data is collected during this preclinical development. In preclinical development, we will try to use, first initially we will use the small animals which are having the body weight of 30 grams to 3 kg body weights. After that, if we get the successful result, then we go for the higher animals like monkeys, dogs, pigs, If we get the better result in the higher animals also, then we'll go for the clinical trials. Clinical trials in clever animal. Clever animal means nothing but human beings. In preclinical studies, in this stage, the toxicity and pharmacological effects of the drugs are evaluated through in vitro and in vivo animal testing. These studies are both short and long-term studies. In preclinical studies, we'll check the both short and long-term studies. That preclinical studies, we must and should follow adhere the rules and regulations of the GLP guidelines. GLP means Good Laboratory Practices Guidelines for to be acceptable for submission to the regulatory agencies such as USFDA, EMEA. For those, we have to adhere to the rules and regulations of the GLP. Here, for the preclinical studies, before going to the animal studies, we have to take approval from Animals Ethics Committee. IIEC, Institutional Animals Ethics Committee, was for conducting of the preclinical trials, we have to take the approval from IAEC, Institutional Animals Ethics Committee. Animals Ethics Committee is an independent body. It is a scientific body. It have the members of biological scientists, generally a pharmacologist or veterinary scientist, 2 scientists from different biological disciplines such as biology and zoology, a veterinarian involved in the care of animals, 1 veterinary doctor, a scientist in charge of animal facility of establishment concerned, and also a scientist from outside the institute, and a non-scientific social awareness member, and also a representative or nominee of CPCSA. In certain cases, if it involves any specialized project, in that cases, a specialist may be co-opted while reviewing the special projects. If we are using any radioactive substances, in those cases, we'll take the help of the radiologist. These are the independent body. They will scrutinize the application and they will give the suggestions regarding the usage of the animals in the clinical trials. As per the norms, first we have to use small animals, rat, mice, mouse, rabbit, guinea pigs. If we got the promising results with the small animals, then we'll go for the larger animals. Larger animals such as mongrel dogs, monkey, chimpanzee, pigs. The ethics committee has empowered, Institutional Animal Ethics Committee is empowered for accepting our proposal or rejecting it or suggesting certain amendments to our application. Their main aim is to check whether the entire process is carried out in a scientific manner or not. And finally, their ultimate goal is to check the safety profile of the new molecule, new entity. The main goal of preclinical studies is to determine the product's ultimate safety profile. Here, by using the techniques, by using both short-term and long-term studies, they will carry out the right compound, right formulation, and right delivery method. This preclinical study avoids the compounds likely to cause problems in clinical trials. Here, we'll pick the right compound. For example, diclofenac sodium, atorvastatin calcium, losartan potassium. While in the drug discovery process, with the diclofenac, they tried with several salt forms: diclofenac potassium, diclofenac calcium, diclofenac magnesium, diclofenac sodium. Among those 4 salt forms, they picked the right compound. Diclofenac sodium shown the better bioavailability, more safety profiles compared with the diclofenac magnesium or diclofenac calcium. Hence, they selected diclofenac sodium. In the similar fashion, in case of the losartan potassium also, they tried with 4 or 5 different salt forms: losartan calcium, losartan magnesium, losartan sodium, and losartan potassium. Among them, losartan potassium shown the promising result with more safety profiles. Hence, they selected. Hence, these preclinical trials will be useful in picking the right compound and also right formulation. We'll avoid the compounds likely to cause the problems in the clinical trials. We'll try with it in the solid dosage forms, liquid dosage forms, and semi-solid dosage form. Among them, in which dosage form it shows the better bioavailability and more safety profiles that they will check. And also picking the right delivery method, whether it is through oral route or parental route or topical route. These final judgment will final conclusions will come in preclinical trials. Based on the preclinical data, the drug discovery has focused primarily on the compound synthesis and target screening. If the drugs succeed in passing all hurdles at the preclinical drug development stage, if we are identifying a right compound, right dosage form, and also right route of administration, all these will be identified in the preclinical level. Then we will submit the application. That application is called as IND. In this, we will submit the data of both animals, short-term and long-term studies. We will submit in the form of the IND. IND means investigational new drug application. After submitting the IND, the regulatory agencies such as FDA approval the process begins only after the submission of the IND application. IND application should provide high quality preclinical data to justify the testing of the drugs in humans. Before going to the human beings, the regulatory authority will scrutinize the IND application, and within the time period of 30 days, it will give the approval. The regulatory authority have the empowered for accepting it or rejecting it or suggesting certain amendments also. Here, maximum within 30 days, within a month, it will give the approval if it is satisfy the results and safety profiles of the new drug molecule. Then they will give the approval for the clinical trials, for the conducting of the clinical trials. For conducting of the clinical trials in India, we have to follow the Schedule Y. A Schedule Y provides the guidelines for conducting of the clinical trials in India. For that, we have to submit the application form 44. We have to take approval from the Drug Control General of India and also an independent autonomous body such as Independent Ethics Committee approval also necessary for conducting of the clinical trials in India. Whereas for the conducting of the clinical trials in Europe and European countries and America, we have to take approval from IRB. IRB means Institutional Review Board. From 2009 onwards, we have to upload the clinical trials data in ICMR clinical trial registry. It is mandatory. CDSCO, Central Drug Standard Control Organizations, will check the facilities of the clinical research organization, whether they are capable of conducting the clinical trials or not. The inspection can be conducted before, during, after the clinical trial is completed. They may conduct the periodic or sudden surprise inspection, whether at any time, maybe before the conduct of the clinical trials, or during the conducting of the clinical trials, or after the conduct of the clinical trials. For conducting of the clinical trials, we have to take approval from Drug Control General of India. It may take 45 working days. Whereas in the case of bioequivalency studies, within 4 weeks, 28 working days. Ethics committee approval for domestic market, we have to take approval from IEC only. For international market is our target, then we will take the approval from IRB, Institutional Review Board. 4 to 6 weeks it takes. Total maybe maximum 14 weeks takes for the taking the approvals for conducting of the clinical trials. For conducting of the clinical trials, we have to follow the rules and regulations specified in the GCP. GCP is the ethical and scientific standards for designing, recording, and reporting of the human research trials. For conducting of the clinical trials, we should abide the GCP guidelines, and also here we have to follow the set of principles for conducting the clinical research. We have to respect 2 themes. Those are respecting the rights of the participants and conducting the research so that the data is accurate and verifiable. For conducting of the clinical trials, first we have to take approval from the participants, voluntaries. From that, we have to take informed consent. It is the mandatory requirement for the initiating or starting of the clinical trials. Informed consent means before agreeing to take part, patient have the right to understand all that is involved in a clinical trial. For that, informed consent form is, we have to, it is voluntary, not forceful. We have to take voluntarily from the participants for the better understanding of the clinical trial. The clinical core team will explain to the participant about the treatment procedure. They will explain in a simple non-technical language, translated in native language of subject. Here, they will explain in their vernacular language, in their mother tongue, they will explain each and everything what has to be happened in clinical trials. A comprehensive information regarding the trial and also benefits of the new therapy over the existing ones and also alternative treatments available. And they will also explain all possible adverse reactions, side effects or toxic effects also, all possible adverse reaction, what they know, they may explain. It may lead to the nausea or vomiting sensation, diarrhea, or certain times it may lead to the coma or death of the patient also. They will explain each and everything. And the participant have the right to withdraw from the trial at any time without giving any reason, because here the participants are human beings, and we must and should respect the human rights also. Here the participation is voluntary, not forceful. After taking the approval, informed consent form, then only we have to conduct the clinical trial. This is the format of the informed consent form. Here, the participant will give his consent for participating in the clinical trial in voluntary manner. After that, we will give the clinical trial protocol. We will submit to the ethics committee for approval. The ethics committee in India, it is called as independent ethics committee, whereas in foreign countries it is called as the institutional review board. In Indian GCP, they mentioned independent ethics committee, whereas in ICH GCP, they mentioned institutional review board. This is similar like as that of the IAEC, Institutional Animal Ethics Committee. Here it is independent ethics committee. It is a board or committee or group formally designed by an institution to review, to approve the initiation and to conduct the periodic review of the biomedical research involving the human subjects. Here it consists of composition of IEC. It consists of a chairman or chairperson, 1 or 2 basic medical scientists, 1 or 2 clinicians from various institutes, 1 legal expert, or retired judge or a senior advocate, and also 1 social scientist or representative of NGO, 1 philosopher or atheist or theologian, or one lady person from the community and also member secretary. Here, this is a scientific body. They know everything about the pharmacology aspects, anatomy, physiology, health education of human beings. And also they have thorough knowledge regarding the drugs which are to be administered to the participants in clinical trials. They will thoroughly check the application, scrutinize the application, and they are empowered for accepting it or rejecting it or suggesting certain amendment to the clinical trial team. They will give the certain guidelines and they will monitor the entire process of the clinical trials. Well, the responsibility of the independent ethics committee is to protect the dignity, right, and well-being of the patients and volunteers. In the first phase, we'll select the healthy volunteers. In the phase 2, phase 3, phase 4, we'll select the patients. Here, they will check whether the sufficient precautionary measures are taken to protect the dignity, rights, human rights, and well-being of the patients and volunteers. And also they will check, ensure a competent review of the clinical trial protocol, and also advise on all aspects of welfare and safety of the participants in the clinical trials. And they will check, ensure the scientific soundness of the proposal, whether the proposal has the thorough references, scientific soundness or not. That also they will check. If any needed, they will give certain suggestions also. Here, after preclinical trials, within a month, they will get the approval for conducting of the clinical trials. After taking the approval from the drug control departments and the ethics committee, and by taking the informed consents from the participants, they will start the clinical trials. First phase 1, it will take months. In case of phase 2, it takes months to years. Whereas in the case of phase 3, it takes years to decades. Phase 4 is continuous ongoing process. One by one, we'll check. In preclinical data, after conducting of the preclinical data, we'll submit the IND filing. After getting the approval, the phase 1, phase 2, phase 3, they will start. First coming to the phase 1. These are mainly the preclinical is also the main purpose is safety. Whereas in the case of phase 1 studies also, the main purpose is safety, primarily meant for the purpose of safety. Hence, for that purpose, in the phase 1 clinical trials normally carried out in healthy adult volunteers. Ikkada healthy means they don't take any concomitant of drug therapy, and also there should be some inclusion criteria. That healthy adult volunteers means they will select 25 to 45 years age. They will select the persons in between the 25 to 45 years age, non-smokers, non-alcoholic, non-drug addicts. They will select. These studies should ordinarily be performed under controlled conditions. Here, controlled conditions means they will select one super specialty hospital and they will admit each participant in a separate rooms. For each room, they will appoint one doctor, one nurse, one clinician, one expert, and also they will make ready of oxygen cylinder, blood, the same group blood, ventilators, each and everything they will maintain. If by maintaining all those, they will take the control over the clinical trials. Hence, it is performed under controlled conditions. The total number of subjects included in phase 1 studies varies with the drug, but usually, generally, they will take in the range between 20 to 100 members. These 20 to 100 members are healthy volunteers. They will select both male and female members. They will select both male and female members. Inclusion criteria is 25 to 45 years age. Exclusion criteria is the females which are at the risk of pregnancy or lactating mother or potent mothers, they will be excluded in the clinical trials phase 1 studies. Healthy voluntaries, non-smokers, non-alcoholics, they will select. Initial dose of the drug is usually low, usually 1/10 of the highest no effect dose observed during the animal studies. Animal studies means nothing but preclinical studies. In preclinical studies, based on the both short-term and long-term studies, they will come to conclusion that this is the safer dose. In that safe dose also, first initially they will select 1/10 of the highest no effect dose observed during the animal or preclinical studies. If, for example, in preclinical, if they found that 100 mg is the safe dose, no effect dose, then they will select 1/10 of 100 mg means 10 mg. 10 mg they will select initially for the phase 1 clinical trials. If the first dose is well tolerated, then the investigation is continued with the administration of the progressively greater doses until some evidences of drugs effects are observed. Initially, they will give 10 mg. After that, step by step, they will gradually increase it 10 to 20, 30, 40, 50, until they will identify some evidences of drugs effects, means some side effects or toxic effects observed, means nausea, vomiting sensation, diarrhea, headache, shivering, like that. If then they will terminate the dose. They will stop the dose. Among the basic data collected before the participate in the clinical trials, they will take the all data. They will check blood pressure, pulse rate, everything, sugar levels, everything, the urine test, blood test, everything they will done. After the dose administration, afterwards also, they will take the all clinical tests they will conduct. And at the same time, they will collect the following data. Which data they collected is its rate of drugs absorption, whether the drug is fastly absorbed or moderately absorbed or slowly absorbed. How is it absorbed? And also concentration of the drug in blood versus time, whether it is detected in micrograms or nanograms, or whether the dose levels, whether those are in the safe level or toxic level. Those also they will check. Rate and mechanism of the drug metabolism and elimination. They will check its metabolites, rate and mechanism, whether the drug is converted into the active and inactive metabolites or not, how much quantity of the drug is absorbed, and how much quantity of the drug is degraded due to the hepatic first pass metabolism. That also they will estimate. And also toxic effects, if any, in body tissues and major organs. They will check any toxic effect of the drug on heart or kidney or liver. Those are on major organs. Those also they will check, and also in body tissues. If the studies demonstrate sufficient merit, and if the order of drug toxicity is very low, and it shows the promising result regarding the safety profile, then the phase 2 studies will begin. Here the first preclinical studies and also in clinical trials phase 1 studies purposes. Their purposes is safety only. If in those 2 steps, if the drug candidate, a new molecule entity shows this safety profiles, then they will go for the phase 2. Here the phase 2, primarily the main objective of phase 2 studies is efficacy. Here we will carried out in targeted patients. Means in the previous case, we will use the healthy voluntaries, whereas in the case of the phase 2 targeted patients, means, for example, if we are conduct the clinical trials for diabetic drugs, then we will take the sugar patients. If we are conduct the studies for cardiological drug, then we will take the BP patients, antihypertensive patients. The targeted patients, for example, if you done the study for the neurological drugs, then we'll take the patients concerned psychological problems patients. They will select the suitable patients, and also the clinical experts also same specializations. For cardiac drug, they will select the cardiologist. For neurological drugs, they will select the neurologist. These studies also performed under controlled conditions. Controlled condition means for each and every patient, for monitoring, one doctor, one compounder, one nurse, one lab technician, and the patient's group, blood, oxygen cylinder, ventilator, everything they make it ready. If any emergency, then they will immediately shift it to the ICU unit, intensive care unit. They will make it ready, ambulances, everything they make it ready, and they will also hire certain experts from other hospitals also for this clinical trials. Then only it is said to be controlled condition. The main purpose is here to check the efficacy. Here, a group of patients with the disease or disorder being studied. Here, to check the efficacy of the drug, the targeted patients will check. And also generally they may enroll 30 to 80, almost 100. We'll select 100 participants in common disorders. In case of rare diseases, we'll select the 30 to 80 members. Generally, they are conducted in the hospitals or clinics approved by the CRO, Clinical Research Organizations, approved by the CDSCO, Central Drug Standard Control Organizations. They may take from up to 48 months or 2 years. They will take for conduct of the clinical trials phase 2. Here in phase 2 studies involved controlled clinical studies to evaluate the effectiveness of the drug in limited number of patients, targeted patients, limited numbers they will select with the conditions for which the drug is intended. Because this phase values the use of patient as subjected side effects or toxicity symptoms that were not shown in the preclinical or phase 1 studies with healthy volunteers may be relieved for the first time. Here, this is the critical phase. Here, there may be chance of risk is more. Hence, they will select the limited numbers. Previous cases, we are selecting the healthy animals, healthy volunteers. But here, we are selecting the targeted patients. Hence, they have to take utmost care while conducting the phase 2 studies. And here, sometimes majority cases and side effects which they are not identified in preclinical or phase 1 studies, they may be observed. Heart palpitations, muscle weakness, numbness, nausea, vomiting sensations, chest pains, diarrhea, abdominal pains, gastric upsets, certain problems, they may arise in this situation. If these are well tolerated, and also clinical experts, specialists in those intended diseases, they will select, they will treat immediately by taking the utmost care to the targeted patients. Limited number of patients with the diseases condition for which drug was developed are treated under close supervision. If they identified that the dose, which one shows the promising result, that dose they will identify in this stage, and if they will check the entire data for its efficacy. After the crossing the which dose, it may shows the side effect. At which dose it shows the safety profiles, they will check that, they will finalize. After finalizing the safe dose levels, then they will go for the phase 3 studies. Here, phase 3 studies are called as these are the better known trials of the safety and efficacy. Here, the main objective is both safety and efficacy. In the first phase 1, objective is safety, phase 2 objective is efficacy, whereas in the case of the phase 3, objective is both safety and efficacy. Yes, both safety and efficacy are the objectives of the phase 3 studies. Here we will include several hundred to 1,000 patients, whereas in the case of phase 1 studies, we will select limited numbers, maximum 20 members we will select. In phase 2, we will select maximum 100, whereas in the case of phase 3, we will select 100 to 1,000 patients. Expanded patient database. We will select a more number of the patients. We will conduct the clinical trials both in a controlled and uncontrolled manner. Here, controlled means as a inpatients in hospital, they will conduct the clinical trials. Uncontrolled means they will conduct outside. As outpatients, they will give the medication, and they will inform to them, come after 12 hours or after 24 hours and give the blood samples or urine or saliva samples. That like that they will inform. That both controlled and uncontrolled. Uncontrolled is also intentionally they will conduct to check the bias of the drug. If whether there is any risk, if it is given, if we conduct the clinical trials in uncontrolled manner, that also they will cross-check. The final ultimate objective is to determine the usefulness of the drug and expanded patient database. Here they will select 100 to 1,000 members, more number, a large number of the patients. Based on that, they will once again cross-check the safety and efficacy levels. Here, group of the patients, they will select maximum, they will select 100 members, 100 to 1,000 members. Maximum number they will try for the regular diseases. In case of the rare diseases, they may select 70 to 150 members. Here they need super specialty hospitals or multi-specialty, multi-centers, because it is not possible to accommodate the 1,000 members, maximum 1,000 members in a single hospital. Hence, they will select multi-centers. They will hire 3 or 4 super specialty hospitals, and in those, they will conduct the phase 3 studies. Hospitals or clinics are possibly several countries. If their intention is to market global market, then they will conduct in 2 or 3 different countries also. They will conduct simultaneously, parallelly. Several dosage strength of the proposed drugs may be evaluated. Here they will check several dosage strength, low dose, medium dose, high dose, with the intention of the children, with the intention of the geriatric patients also, they will check. For the children, the low dose is also less, whereas in the case of older patients, geriatric patients also, the dose may be less. Hence, they will check with several dosage strength. After getting the sufficient information, the overall benefit risk relationship of the drug, they will identify, and after getting the conclusion, then they will file a complete NDA, new drug application, to the regulatory authorities. The regulatory authority will check the NDA, new drug application details regarding the both benefits risk relationships, safety and efficacy levels. After submitting the NDA, FDA or any regulatory agency review and approval after completion of the clinical trials that shows satisfaction of the medical community that the drug product is effective in all parameters and is reasonably safe, demonstrated by the animal and human studies. And whether it is effective in all parameters. All parameters means quality, efficacy, safety, purity. Quality, purity, efficacy. Along with them, it should be reasonably safe. It should also fulfill the criteria of safety also, as demonstrated by the animal and human studies. Animal studies means nothing but preclinical studies. Human studies means nothing but clinical trials. While conducting the animal studies, we must and should abide the rules and regulations of the GLP, Good Laboratory Practices Guidelines. Whereas while conducting the human studies, we have to abide the rules and regulations of the GCP, Good Clinical Practices Guidelines we have to follow. After successful results, the regulatory authority will give the approval for marketing of the new drug. Then these are called as the post-market trials. Phase 4 clinical trials will run. A post-marketing surveillance study. These phase 4 studies are also called as the post-marketing surveillance study. After NDG is submitted and before approval to the market, the product is obtained approval from FDA, manufacturing scale-up activities will takes place. Because up to phase 3, we will use limited number of the dosage forms for clinical trials. Generally, those may be developed in the lab scale. Hence, to go for the large scale while going before to the marketing level, then they will go for the full scale production from lab to pilot plant to industrial scale. They will develop the product and they will optimize the product, modify slightly as a result of data obtained during the manufacturing of the scale up and validation process. Here, the phase 4 studies also will continue even though after getting the approval from the drug regulatory authorities. Those were generally it takes yearly years. It takes. They will, if the drug is successful in market, then it will sustain in the market for a prolonged period of time. If there is any problem, if they observed any unwanted side effects, if they are noticed after entering into the general public in phase 4 studies, then they will bring it to the notice of the regulatory authorities. Then the drug may be banned. They will cross-check everything, its benefit and risk relationship, and they may suggest it for other indications, or certain times they may ban the drug. At that time, the pharmaceutical company withdraw from the market. Here, this entire process is a very risky, time-consuming and tedious process. During this drug discovery process, if you try for 10,000 compounds, only after 15 years, we may get approval of single drug, one drug. Here, if you try for 10,000 compounds, then regulatory approval, there is a chance is 1, 10,000 is to 1. There is for 10,000 trials, there is a chance of getting success is only 1. After FDA grants market approval of the drug product, even though drug product development may also continue. The drug product may improve as a result of equipment supply and also based on the market demands. After during the phase studies, sometimes the patients may feel some inconvenience regarding the color of the drug or its organoleptic characteristic features. Sometimes some people they don't like its taste or odor. In those cases, that R&D team, they may modify its color, flavor, taste also, based on the market demands also. Sometimes the approved drugs are also banned due to the unwanted side effects. When the side effects are unacceptable and safe alternatives are available, for example, analgin and rofecoxib, a more safe alternative is available, then the first generation drugs will be demolished and the new drugs will enter into the market. When superior medicine with few side effects are available, the previous one will be fade out. When side effects are more dangerous than the diseases, in those cases also drugs will be banned. The use of those medicines should be discouraged. Here there are certain examples. Those examples are analgesic. This is indication for the purpose of analgesic, but it causes the bone marrow depression. In case of the cispiride, it is mainly for the purpose of acidity and constipation, but it causes the irregular heartbeats. Whereas in the case of furazolidine, it is intended for antidiarrheal, but it leads to the carcinogenic, cancer-causing. Nimesulide, nice tablets. These are painkiller or fever. Indication is for painkiller or fever, but they cause the liver damage, hepatotoxicity. Hence, like that piperazine, it is antihelminthic, but it causes the nerve damage. Phenylpropylamine, it is intended for cough and cold, but it leads to this heart attack, strokes. And also nitrofurazone, it is antibacterial topical cream, but it leads to the carcinogenic. If the drug intention purpose, we are bring it into the market with some intention, but it shows the unwanted side effects. And here the risk is more than the benefits. In those cases, the drugs will be banned. If the drug is success without any unwanted side effects, then it will sustain in the market for a prolonged period of time. Even after years also, after entering 10 years or 20 years also, a post-marketing surveillance will continue, done to assure the efficacy and safety of the drugs after its marketing. Marketing also, post-marketing studies will continue for until it will retain in the market. These phase 4 studies will be continued. India nowadays, for the clinical trials, majority of the global trials are now running in India also. Coming to the last 15 years scenario, from 2005 onwards, now the number of clinical trials participants and also trials also increasing enormously. And also the number of patients, number of trials also very significant number. Now the India become the gateway to the clinical research in compared with the foreign countries. The main advantages of India to become the hub for the clinical trials is well-developed industry with a strong manufacturing base and well-established R&D infrastructure. We have the rich biodiversity. We have several sources of drugs, plant sources, animal sources, mineral sources, marine sources also available. There is rich biodiversity in India. We have Himalayas herbs, we have Nalamada shrubs, and also Nilgiri forest, the sources also we have, marine sources we have Deccan Plateau, we have the Himalayas mountains also. There are rich biodiversity. There is more sources of drugs, mineral, marine, and also biological sources, plant and animal sources also available. And also India is the third largest English-speaking scientific and technical manpower in the world. And also lower cost of scientific manpower, highest intellectual capital per dollar. We have the lower cost of scientific manpower in the world, Indians. And also investigations such as animal testing and pathological tests are so cheaper in India compared with in US and European countries. Hence, the clinical research activity in India costs 30 to 40% lower than in Europe or US. Hence, now the India become the hub for clinical trials due to the availability of the strong manufacturing base and also hospitals and also manpower and also technical people with the specializations of SAS and pharmacovigilance clinical trials. Now, majority of the members are recruited in the clinical trials, the pharmacovigilance section also. With this, I conclude the design and development of the drug development process. I hope the purpose of organizing this FTP is fulfilled and seek the same response in future also. I request my fellow teaching faculty, please, with this knowledge, please inoculate the creativity and innovation ideas in the minds of students. If you really inoculate the creativity innovations in the minds of the new generation students, then it will give the fruitful generations to our India. Then India will become the superpower. Thank you. Thanks to one and all. If anybody have any queries, please ask.
Thank you, sir. Thank you for your insightful and enlightening presentation, sir. Your presentation has broadened the understanding of the regulatory requirements for the drug development. I am sure that this session will be very helpful for the participants. Now I request the participants, if they have any queries, the floor is open for the queries.
If anybody any queries, please ask. One of the person asked in chat box, what is the scope of artificial intelligence in regulatory screening and drug discovery process? This is the good question. Artificial intelligence is a now upcoming scenario. There is upcoming scenario. Now, actually, this drug discovery process, it is the time consuming and tedious process. Generally, it takes 12 to 15 years, maybe with the help of the artificial intelligence. In the coming days, the time may be reduced. Everybody trying by utilizing the artificial intelligence, the number of trials, they are trying to reduce the number of trials. Maybe in future, the time will be reduced, and also number of the attempts also may be reduced with the help of the artificial intelligence. In chat box, all of you responding with positively. Thanks for all.
If anyone have any other queries, you can ask Dr. V. Sai Kishore garu. On behalf of VV Institute of Pharmaceutical Sciences, I am very thankful and grateful to Dr. V. Sai Kishore garu. Sir, as a token of our gratitude, please accept this certificate of appreciation.
Thank you, sir. Thank you, sir.
Thank you, sir. Thank you for your valuable time, sir. It was a very informative session, sir.
Okay. Thanks to Dr. Lakshman Rao Garu, Principal, VV Institute of Pharmaceutical Sciences, and thanks to office bearers of APTI, Andhra Pradesh branch. Thank you. Thanks to one and all. Hello. Shall I leave, sir?
Yes, sir, you can leave, sir. Thank you so much, sir.
Good.
Dear participants, I have sent the day 2 attendance forms in the chat box. You can all fill the Google form and leave. Thank you so much. Yes, sir.
farm.